So this is some joint work with not only my advisor, but also an undergraduate student who also made the trip, Yang Jin. And the title, as was said, is Assessing a Spatial Boost Model for Quantitative Trait GWAS. So to get started, I'll just tell you what I mean by quantitative trait GWAS, our problem description. Then I'll tell you our solution to the problem, the spatial boost model. I'll describe that. And after I define our model, I'll tell you what it is we want to assess about it and go into more detail through our simulation setup for our simulation study. Then we'll take a look at some results, and then I'll wrap things up in the conclusion. So the problem we're trying to solve, it's very simple. Simply stated, but very challenging. So GWAS is an acronym. It refers to Genome-Wide Association Study. And basically what we want to do is we want to take a set of features which are numerically encoded pieces of DNA from a number of different people, and then try to find the statistical relationship, if it exists, between the set of numerically encoded DNA and some response variable. So for us, since we're doing quantitative trait GWAS, we're interested in quantitative traits like blood pressure or body mass index or birth weight, things like this. So these numerically encoded pieces of DNA I'll call variants. If you're familiar with this problem, you may have heard the term single nucleotide polymorphism or SNP. They have a lot of different names, genetic markers. I'll stick to variants for now, though. So it's a simply stated problem, but it's challenging because it turns out for any one individual, we have a large amount of these variants, typically on the order of 500,000 or more. It's only growing as technology gets better. And for a typical GWAS data set, we have millions of these pieces of DNA, these variants. But maybe if we're lucky, only 5,000 or so samples. So it's a large piece, small n problem. And if you try to do a usual multiple linear regression model, you won't be able to find the estimate for your model parameters, the ordinary least squares estimate, because it's an ill-posed problem. So we have to do something to overcome this curse of dimensionality. What's traditionally done is researchers, geneticists, they take one column at a time, one variant at a time, and fit a simple linear regression. So they regress the response variable on one column at a time, and then they run a hypothesis test to see if the slope of the regression model is statistically significantly different from zero. And then they do this for each variant. So they build P, simple linear regression models. They conduct P, hypothesis tests. And they come up with a p-value, or a measure of association for each variant. And they decide on a genome-wide significance level. And then they try to set the threshold and look to see which variants are statistically significant over the whole genome. So this typical approach, after they find a significant variant, they'll then look to see on the genome where does this variant lie. Maybe they can find a gene close by that might explain something about the trait. And if they can do this successfully, it's great, right? Because if you can solve this problem, the whole motivation is one day we can have personalized medicine. You can go to your doctor, the doctor can take a sample of your DNA, maybe understand something about what is causing you to have a particular trait. Maybe we can develop some better medicine or cures for some diseases. But in any case, the traditional way of tackling this problem, it's too simple. In the sense that we ignore possibilities for joint effects. We're not analyzing the variants jointly. We don't look for interaction effects. We only fit these very simple models, one at a time. So what we would like to do in this work is to try to come up with a way to fit a more representative model, taking a hierarchical Bayesian approach, do some variable selection, and see if we can do this in a computationally feasible way. Right. So to people in this field who do GWAS, they recognize that there are other ways to solve the problem. You can use penalized regression, or you can use a Bayesian technique, but it's very computationally intensive. And so rather than use these methods, they still stick to these single association tests. So what we would like to do is try to bring something new to the table where we actually have a model that's more powerful, it's more representative, we model the data jointly, but also one that we can fit in a reasonable amount of time. So we have a couple new ideas here. So here's the spatial boost model, as we call it. It's a hierarchical Bayesian model for Bayesian variable selection. A lot of it is very standard for GWAS, but we have something new. So first of all, if we have our data set with n individuals and p of these variants, we model the trait, the quantitative trait, conditional on some model parameters beta and a variance tau squared. As just being normal, with expected value equal to the linear combination of your variance with the variance tau squared. We have a continuous spike and slab prior distribution for the model parameters beta j, so conditional on an indicator variable theta j and another variance term sigma squared. The model parameter beta j will have a normal distribution with expected value 0 and a variance equal to this term here. So if the indicator variable theta j is 0, it will mean that the variant is not associated to the trait. And so for the variance term for this prior distribution, this term of kappa drops out, and we're left with sigma squared. So a priori, we expect sigma squared to be a small number. If theta j is equal to 1, we see that sigma squared gets augmented by a factor of kappa. So we would want kappa to be a large number so that there's a separation between the spike and the slab here. So here's where our first novelty appears. We model the prior probability of association for a variant as, so indirectly through this linear combination of a gene boost term and some other parameter C naught. So I mentioned earlier, the traditional approach to this problem is to analyze each variant independently one at a time to look to see which variants are genome-wide significant, and then to look in the area around that variant to see if there are any genes that might explain something about the trait of interest. The reason people do this is because it's uncommon to know the biological function if it exists of one particular variant. It's much more common to understand something about the gene. So we try to include that in our model right from the start. So we incorporate this through a gene boost term, which I'll define on the next slide. So we have some weights in this WJ transpose phi term. So we have gene weights for each variant, parameterized by phi, which encode something about the spatial distance of a variant to genes on the chromosome. And then we have this other term R, which encodes the relevance of a gene. So the full gene boost term is WJ transpose phi dot R. It's a dot product, and it captures, it's like a weighted sum of these gene weights and their relevances. So we normalize this term so that the maximum value is 1. Then we have this parameter C1, which controls the scale of this gene boost term. So you can really think of it as the maximum log odds increase of a particular variant being associated to the trait coming from the gene boost term, this C1. So C naught then, if there is no gene boost, so if this term was actually 0, or if a variant is in a region of the chromosome where there are no genes close by, C naught captures indirectly the prior probability of association for a variant in such a situation. For the variance terms tau squared and sigma squared, we have the conjugate prior inverse gamma with hyperparameters nu1, lambda1, nu2, and lambda2. So as we said earlier, sigma squared we would expect to be small. So for example, maybe 10 to the minus 4th. We can choose the hyperparameters here to reflect that a priori assumption. For tau squared, we can choose our hyperparameters to be non-informative and try to help let the data decide what value would be appropriate. So here's a little more detail about the gene boost term and our gene weights. So the weights themselves are defined in this way. And the equation is here, but it's much easier if I just start from the picture. So basically what we do is we center a Gaussian curve at a particular variant. So if you can see here, I have a variant A in red, and I have some genes in green. So we center a Gaussian curve at a particular variant, here variant A, and the curve will have a standard deviation equal to this phi, and we call phi our range parameter. So then after we pick this phi, and we have our Gaussian curve centered at a particular variant, we just calculate the area under this curve from the transcription start site of a gene to the transcription end site of a gene, and use that as our weight for a particular gene. And the relevance is this R vector We have a few ideas of how to define the relevance of a gene. There are some text mining algorithms out there that search the GWAS literature and build a score based on the co-mentionings, the number of co-mentionings of a gene and a particular trait. We could also choose a non-informative vector, just of all ones, let every gene be relevant. Okay, so now that you know the model, let's talk a little bit about how to fit it and how we like to fit it in a computationally feasible way and how we'll assess that. So first of all, we have to define all of our model tuning parameters. There's no shortage of them. We have a lot here. I've talked a little bit about how you might choose these hyperparameters. We have a method of choosing phi as well. I'll talk a little bit about that later. What we really want to understand, though, is the effect of misspecifying a value of kappa and misspecifying a value of C1. So kappa controls the separation between the variants of the spike and the slab paste. It plays a very important role in our variable selection. C1 measures the strength of the gene boost term, and that's the whole crux of our spatial boost model. So we really want to understand what happens when you vary these, so do a sensitivity analysis. But once we define all the model tuning parameters, the next step is to actually fit the model. And if you try to just fit a Gibbs sampler, you try to run a Gibbs sampler with all of the markers, all of the variants, it's very, very slow. The solution we've come up to overcome that obstacle is this. So we have an expectation maximization algorithm. We run this on our data set, and we build the conditional posterior probability estimates, so conditional on the final results of the EM algorithm. We rank, we sort all of the variants based on these values, and we remove the bottom quartile. So it's a filtering step. So we continue doing this step, this EM algorithm. We fit the EM algorithm, we sort the variants based on this quantity, and we remove the bottom quartile until either the number of markers or variants is small enough to handle feasibly with a Gibbs sampler, or until the predictive accuracy of our model deteriorates beyond a certain point. So I won't go into all the details for the EM algorithm, there's not much time, but I can just tell you we use an E step on theta, we treat theta as a latent variable, and then we use an M step on beta, sigma squared, and tau squared. Once we have our reduced set of variants, we can then run our Gibbs sampler and compute our final estimates of the posterior probabilities of association with our centroid estimator here. So we have a way to further speed up this EM filter by taking advantage of a computational trick with the M step on beta. So I'll mention that too as well, because we want to understand how this trade-off will affect the performance of the model. So gaining some speed, but maybe losing some performance. So we have this equation to update beta in the EM algorithm. And the most important part to take away from this slide is that up here, we originally had this P by P matrix. You have to take the inverse of a P by P matrix. It turns out if you use an SVD approximation to X, so if we write X equals UDV transpose, and in particular, if we take a truncated approximation, so we only use, let's say, K singular vectors, then using the K-lith variant of the matrix inverse, we can actually replace this inversion of a P by P matrix with the inversion of a K by K matrix. So it's a huge speed up in the computing time. But again, we want to understand what is the effect of choosing a smaller number of singular vectors on our model performance. So let's talk about how we actually assess these things. So for completeness, I have all the details for the simulation study up here, but I'll just tell you briefly the general idea. We want to understand how the model performs across all the genome. Right? So to do this, we actually take one chromosome and we randomly sample contiguous blocks of 1,000 variants at a time. So we generate 100 data sets in this way. So we have a real data set provided by a consortium over in the UK, the Wellcome Trust Case Control Consortium. And we take this data set and we randomly sample 100 individuals and then contiguous blocks from these 100 individuals of 1,000 variants. So we do this 100 times and we get our 100 replicates of X. And then we set some values for tau squared, sigma squared. So tau squared is 100, sigma squared is 10 to the minus 4. We set values for phi using our proposed criteria. If there's time, I can talk a little more in detail about that. If not, we have a paper up here in the archive that goes into more detail. For this work, we just consider a non-informative gene relevance vector, so a vector of 1s. And then we set our C naught to be the logit of 10 divided by PS, where PS is the number of variants in our S replicate data set. Then we simulate theta so that we have 10 variants that are actually associated with the trait. And then we simulate beta and Y. And finally, we're going to fit the model in a variety of settings and assess the model performance using AUC, the area under the ROC curve. So using our knowledge of the ground truth here, we can actually construct the ROC curve and then compute the area under it. So there are 3 things we really want to assess. First of all, we want to understand how the model performs in a variety of different settings. So in particular, we consider 6 combinations. First, when there is no gene boost, and then when there is a gene boost. And then, so we make a connection to the concept of heritability. So the heritability of a trait is defined as the proportion of the variance of the trait that is explained by the variance in the genotypes, or these variants. So we have a way to choose kappa to control an approximate heritability of the trait Y. So we consider a low heritable trait, medium heritable trait, and a high heritable trait. So then we fit our model using the original X and also SVD approximations of X with a mean squared error tolerance of 1%, 10%, and 25%. For the second simulation study, we want to understand what happens as we vary kappa with all the other tuning parameters ideally set. For the third simulation study, we want to understand what happens as we vary C1, this scale for the gene boost term. So let's take a look at some of the results real quick. So we can see what we have here are some box plots. On the left, in every subplot, are the AUC values, a box plot of AUC values when we run the traditional analyses, the single association tests. For the rest, we have these percentages, 0%, 1%. The percent indicates the tolerance we used on the mean squared error in our approximation to X, the SVD approximation. 0% means we use the actual matrix X. The rest are all SVD approximations. The 2 box plots that we have in each subplot for our method, the first is what we get if we actually just use the final conditional posterior probability estimates from the EM algorithm, the EM filter. The second one is what happens when we actually use our final posterior probability estimates from the Gibbs sampler. So the salient points here are we actually see not much of an improvement, not a significant improvement when we use our final estimates from the Gibbs sampler as opposed to using just the final estimates from the EM filter. You can see as the heritability of the trait increases, we see a decrease in our model performance as we take a coarser approximation to X. This is because more and more of the variation in the genotypes is explaining the variation in the trait. Of course, when there is a gene boost term, this is a sanity check, we perform very well. For the second simulation study, we see it's actually better to choose a relatively smaller value of kappa, even when the true value of kappa is larger. And we suspect this is because it's difficult to model simultaneously strong effects and weak effects with a large value of kappa. For the third simulation study, we see that our model is robust to misspecifications of C1 when there really is no gene boost term, but otherwise sensitive when there really is a gene boost term and you misspecified C1. Okay, so just to wrap up, the real important points that we've seen in this analysis is that, first of all, we have this fast way through our EM algorithm and the SVD approximation of fitting our spatial boost model for these quantitative traits. We've seen that when we use our final results from the EM filtering step, we actually see roughly the same level of performance of that when we use our final results from the Gibbs sampler, which is much slower. That's nice. When we actually do have a gene boosting effect, as we have it defined in our hierarchy, we can recover these signals even when we use a coarse approximation to X, large tolerance for mean squared error. And for future work, we're considering a more thorough simulation study, and even for real data, analyzing chromosomes in blocks to speed up the process even more. All right, so that's it. Thank you all for your kind attention.